Contract sessions at the New Zealand Society of Animal Production have been a feature of the conference for a long time, and the idea behind them is it's the opportunity to look at one topic in a bit of detail and sometimes to involve speakers who wouldn't normally present at the conference to be part of that. So this session is on genetics. Beef and Lamb New Zealand Genetics have kindly sponsored the session, so a big thanks to them, especially since they have their annual awards on tonight and have to get down to Invercargill in order to do that. So the way that the format of the contract you'll see in the program is that each of them gets 15 minutes. What we'll do is they'll talk for 10 minutes. We'll allow a couple of points of clarification, but have a panel discussion to ask questions at the end. The exceptions being Eleanor and Mark have to leave immediately after that, so we'll allow just a couple more questions for them, and then we'll have a stand-in to answer any of the awkward questions which they would prefer were asked during the panel discussion. In 2009, we had a contract session at NZSAP called SIL the First 10 Years, and obviously it celebrated and looked at the progress that had been made in SIL Sheep Improvement Limited, delivering genetic progress to the sheep industry over a 10-year period. Time has rolled on. We're 6 years down the track, and there's a lot that's happened. And we are looking at SIL, but we're also taking a wider look at the research that's been done and the investment that's gone in through a number of projects where levy payer money, government money, and third parties have invested in genetics. So it's things that have been applied since then and are in the process of being applied and what's going forward. So it should be a great session. So you didn't come to listen to me. I'll hand over to our first speaker. So Eleanor Linscott's going to present our first one. She was the Ovita Consortium Manager, and she's now the Science Manager in Beef and Lamb Genetics, and she's talking about Ovita 2008 to 2012. 13. Thanks, Eleanor.
Thank you, Neville. Why do I feel like we've been damned with faint praise? Sure we haven't. So, as Neville has just said, I'm the Science Manager with Beef and Lamb Genetics, but with Ovita I was the Consortium Manager. So this really is the whistle-stop tour of Ovita. Ken is talking as well, and he's going to go into a lot more detail. So, basically, the 30th of June 2013 officially marked the end of Ovita after 10 years of investing in sheep genetic research. In 2003, Ovita had a wide focus with sheep genomics, reproduction, muscle biology, parasites and wool. Ovita had 3 shareholders. There was Meat and Wool New Zealand, AgResearch and Wool Equities. And industry investment from levy payers was leveraged with the Foundation for Research, Science and Technology. Ovita restructured in 2006 to be focused mostly on genetics, sheep genetics and diagnostics. So Ovita, from the years 2008 to 2013, which is what I'm going to be talking about today, was basically a partnership between Beef and Lamb and AgResearch, and industry levy funds were leveraged with investment from MB, the Ministry for Business, Innovation and Employment. And there were other investment from third parties as well. So the objectives of Ovita was to strategically target sheep genomic and biotechnology research opportunities, to develop outputs for application on farm for the benefit of New Zealand sheep industry, and to assist with the maintenance of research capability. The purpose was to fund the generation of new knowledge to deliver commercial outcomes for the benefit of the New Zealand sheep industry. So, along with that, the Ovita stakeholder expectations, so this is our levy-paying stakeholders, were to deliver products in the near term which deliver significant benefits to the farmer's bottom line profit, to deliver a product with value and at an affordable cost, and to justify that levy investment to farmers via Beef and Lamb New Zealand, and to provide tests for a range of breeds, to make the delivery mechanism commercially sustainable, and to maintain that scientific capability. So the Ovita achievements in the first 5 years of investment, there were single gene tests and microsatellites. So basically the tests that came out were Myomax, Loinmax, Inverdale, Wormstar and iScan. And iScan was a test for microophthalmia in sheep. And also in that time, the first sheep DNA parentage test was released, and this was the shepherd test. Blood tubes and FTA cards were the sample type. So FTA cards is the bloods on a card. Right? So from 2008 to 2013, the Ovita achievements were tissue sampling and extraction methods developed. So this is DNA extraction, tissue samples from on-farm collected and extracted in the lab. And genomic selection, SNP chips for sheep developed, the sheep 50K and the sheep 5K, and Ken Dodds will go into more detail on this. Myomax and iScan SNP-based tests were developed, and the Carla test developed. And there was second generation DNA parentage test released. So this is the Shepherd Plus, which is a SNP-based parentage test. But Ovita was not only about DNA tests. We had additions or changes to SIL, which was things like the update to survival, adult ewe traits, DAGs and fly strike. There were also increased FE phenotypes and genotyping, increased worm FEC numbers, increased ewe fate code records in SIL, and increased the number of flocks that we were working with to over 150. In summary, Ovita achieved many things and not just DNA tests. The 2013 year was a successful year for Ovita with the release of the 5K test and SIL blending. And this would not have been possible without the help of many ram breeders throughout New Zealand and the number of people in organisations working on and funding the projects. So in Ovita capability, there were 75 ag-research staff were involved working on Ovita projects at any one time, and this included field staff, lab and analysis staff. So Ovita trained 3 postdocs, 4 PhDs, 2 MSc students and 1 DipSci. And of the 4 PhDs, there are 2, both Natalie and Gemma are still working in sheep research in New Zealand, and the MSc student, Catherine, is also still working in sheep genetics. And we also had over 10 visiting PhD students from abroad. In the research flocks, we had 337 flocks and nearly 220 operations contributed to Ovita by providing DNA samples, records, and most significantly, their time. So, the thank you to all the groups that were involved with Ovita during that time. I've listed them all there. But the big thank you also goes to the Beef and Lamb New Zealand levy payers and the Ministry of Business, Innovation and Employment. Thank you.